Hi guys, I'm Ashish and you're watching me on Medical India. So guys, today's lecture is very special and very kind of versatile, you can say. In AIIMS PhD entrance, ICMR, JRF, DBT, JRF, all the examinations were aptitude, and basically this cell bio becomes, or general biology becomes the main part, then it comes into existence. So guys, what happened? Last month I have started the lecture series on the AIIMS PhD entrance, and it is not the lecture series which is going to benefit you on the AIIMS PhD entrance only. So please guys who are preparing for CSIR JRF, ICMR JRF, DBT JRF and GATE, please watch this lecture series, and this is going to benefit you in many ways. In ICMR JRF you are going to get 25 questions from my lecture series, okay, and in DBT JRF same 20 questions aram se you are going to get in the like DBT JRF exam, and in the like AIIMS PhD entrance I can claim you will get 15 to 20 questions out of 30 in the aptitude part. So guys, this is very important lecture, as you have like already understood about this. Okay, so guys, let's start the topic with the very first lecture, and it is based on the Nobel Prizes. Well, Nobel Prizes becomes very important if you are going to get the aptitude based questions in any examination. Then Nobel Prize is asked many times. In the DBT oftenly asked, in the ICMR JRF oftenly asked, in CSI it may ask, but this is not the thing that can be asked. But in AIIMS PhD entrance every time Nobel Prizes are asked. So you have to cover the 5 years Nobel Prizes of Physiology and Medicine. So at David Julius, Ardem Patapotian, so for their discoveries of receptors of temperature and touch, got the Nobel Prize in Physiology and Medicine in 2021. So this is the slide which is showing you the Physiology and Medicine Nobel Prizes. So in 2020, Harvey J. Alter, Houghton and M. Rice, they got the Nobel Prize in Physiology and Medicine for the discovery of hepatitis C virus. And in 2019, Kylan, Peter and the Semenja, they got the Nobel Prize for the like kind of discovery on how cells sense and adapt the oxygen availability. Okay, so in 2018, Ellison and Honjo, everyone, they are the like famous scientists. So they got the Nobel Prize for the discovery of cancer therapy by inhibition of negative immune regulation. In 2017, in the circadian rhythm, the Nobel Prize was given to Hall, Rosbach and Young. Okay, so please remember guys, there is no concept, or you can make the trick if you want to remember these kind of 5 years Nobel Prize in Physiology and Medicine. So guys, these 3 becomes very important in many examinations. Sometimes they ask the 2020 Nobel Prize in Chemistry, which was given for the genome editing in chemistry. You can see here, who were they? Charpentier and Dodena. So they got the Nobel Prize in 2020 in chemistry field in the genome editing. And 2014, everyone knows there is a movie on the Kailash Satyarthi who was awarded Nobel Prize in Peace for their outstanding work in the kind of field of child education and to stop the child labor. Okay, so they got the Nobel Peace Prize. Okay, Abhijit Vinayak Banerjee in 2019 got the Nobel Prize in Economics. Okay, with 2 others. And why? Because they have the idea to like alleviating the global poverty. So these 3 you can remember. Okay, so let's go ahead and discuss hormones and deficiency. In many examinations, hormones and deficiency becomes very important. So what happens? Hypothalamus secretes the hormone and many hormones basically, and it basically gives their hormones to the posterior pituitary. Okay, so let's discuss the pituitary gland. Why? Hypothalamus has no own secretion system. It secretes, transfer their secretions to the pituitary posterior gland, and then posterior gland gives the kind of thing to the other circulation part of the body via circulation. So posterior pituitary releases 2 hormones basically, oxytocin, which is peptide in nature, antidiuretic hormone, that is peptide in nature. You can take a screenshot of this lecture like slide, or you can pause the video and go in detail. So okay, so let's go ahead with anterior pituitary, which is going to secrete the growth hormone, and it is protein in nature. It is very important. It is very important. These 2-3 slides are very important. Okay, anterior pituitary also is going to secrete the prolactin, and it is protein in nature and it stimulates milk production and secretion. Okay, follicle stimulating hormone LH, these 2 are basically the glycoproteins, and they are, you all know, are the like kind of germinal, you can say, secretions, hormones, germ hormones. Okay, thyroid stimulating hormones are glycoproteins, and they stimulate the thyroid gland. So adrenocorticotropic hormone ACTH is the peptide, and it stimulates the adrenal cortex to secrete the glucocorticoids. Okay, so please remember this. Thyroid gland is going to secrete the triiodothyronine and the thyroxine, T3 and T4 basically. T3 is amine and T4 are amine, and they are going to stimulate and maintain the metabolic process. Various processes they are involved in. Okay, so calcitonin is a peptide which is going to lower the blood calcium level. And what is it? It's like kind of analog. The analog is the parathormone. See here, parathormone, PTH. Okay, parathyroid hormone or parathormone you can say. It is a peptide in nature and raises the blood calcium level. So pancreas are going to secrete the insulin and glucagon from the alpha cell, and beta cell the insulin. So they are protein in nature and lowers blood level and raises blood level. They are the antagonist. Okay, so adrenal medulla basically is going to secrete the hormone, that is epinephrine and norepinephrine, and they are amine in nature, or they are going to raise the blood glucose level, increase metabolic activity, and constrict certain blood vessels. Okay, adrenal cortex basically is going to secrete the glucocorticoids and mineralocorticoids. They are steroid in nature. Okay, please remember, adrenal is going to secrete the both amine and steroid. So you can remember the function of glucocorticoids, that is increasing the glucose level. Okay, so you can like think about it in the context of the insulin, which is going to lower the blood glucose level. Okay, so this is the antagonist. So this is asked many times. Mineralocorticoids are steroids in nature, and that promotes the reabsorption of the Na+ and excretion of K+ in the kidney. So this is very important. Gonads, testes and ovaries, no need to tell, but they are going to secrete the androgen and estrogen respectively, and they are steroid in nature, and basically they are having the role in the development of the like kind of, you can say, the primary and secondary character. Okay, so pineal gland is going to secrete the melatonin, which is amine in nature, and it has the role in the circadian rhythm and many biological cycles rhythms. So guys, taxonomy, it becomes very important part. Many times questions are asked from the taxonomy, and this is enough. This slide is enough. Taxonomy, if you are going to study, I have the full lecture series on taxonomy, and this is like kind of 20-25 hours. But this is enough at the level of like aptitude part. Okay, alpha taxonomy, it is concerned with finding, describing, and basically the identification part, you can say, is the describing or description part of the organism basically is here in the alpha taxonomy. In beta taxonomy, the identification of the natural group and the biological classes. Okay, biological class means classification part is going to happen in the beta taxonomy. First, what happened in alpha taxonomy? The description of the organism was, or the plant was given, and in the beta taxonomy, identification and classification was done, and in gamma taxonomy, the study of evolutionary relationship or phylogenetic relationship was established. Okay, so you can here think that species are the like kind of subsay smallest taxon, and then genus, then family, then order, then class, then phylum, then kingdom, and then domain. So please remember this. Domain you can leave, but the 7 are very important. Sometimes what happens, order are asked. Okay, so please like kind of order you can remember. So cell signaling information, cell signaling is a vast subject, as you all know. It is a kind of subject only. Okay, so signaling molecule and the pathways corresponding to them are given here. So alpha subunit of G protein is associated with the activation of adenylyl cyclase. So insulin growth factor is going to have function in the receptor tyrosine kinase pathway. Erythropoietin in non-receptor tyrosine kinase pathway. CAMP, which is secondary messenger, and epinephrine are having the role in protein kinase A pathway activation. Okay, rhodopsin is going to mediate the G protein pathway, and PIP2 is going to contribute in the IP3 DAG pathway. DAG is going to activate the protein kinase C, and RAS is going to be the target in the MAP kinase pathway. So please remember these things. These things may be asked in the examination. So pedigree becomes very important. You can go through all the symbols. No need to explain. Male, female, marriage means mating, and contiguous mating. Okay. Then the Roman numerals symbolizes generations. Okay. And you can see identical twins and fraternal twins females, affected male and females, okay, carrier, decreased male and female, deceased male and female means aborted, and unknown sex. Okay, so please go through it. Then we are having the trick how to identify the autosomal dominant, autosomal recessive, sex-linked recessive, and X-linked dominant. So guys, here given, okay, if all the like generations have the like kind of dominant part, then that is the autosomal dominant, okay, and it is distributed in all the sexes. Okay, cannot be recessive as two affected patients could not have an affected offspring. Parents must be heterozygous. Second, C, autosomal recessive cannot be dominant as two unaffected parents could not have an affected offspring. Parents must be heterozygous. What happened? They are skipping the generation, so it is a recessive. In all the generation, then dominant. And X-linked is seen in a kind of pattern in a single, you can say, gender. Okay, so here sex-linked cannot be confirmed. 100% incidence of affected daughter from an affected father suggests X-linked dominant. Okay, affected father, affected daughter from the affected father. Okay, so this is the X-linked. And then X-linked recessive, it has sex linkage cannot be confirmed. 100% incidence of affected sons from an affected mother. So the opposite of that, you can say the X-linked dominant. What happened there? Father was affected, so the daughters were affected. Here mother is affected and the sons are affected. Okay, so it suggests the X-linked recessive. So come to the RNA. RNA is of three types, everyone knows: messenger RNA, ribosomal RNA, tRNA. Okay, so messenger RNA is also kind of unprocessed, you can say, hnRNA. The unprocessed form is the hnRNA, and it has the uracil basically in all the RNAs. And then what happens? Here comes the subunits. Subunits are many times asked in the examination. So you can remember that the prokaryotes have the 50-30, and that makes the 70S. S is the sedimentation coefficient, or you can say surface area basically. So eukaryotes have the 80S, and 60S and 40S constitute the 80S. So you can see here that 40S may 18S, and RNA is there with 33 amino acids, and with 33 proteins, and 28As, 5.8S and 5S are going to constitute the 60S, and 50S may 23S, 5S and 5S are going to constitute the 50S. 30S is constituted by the 16S RNA. So guys, please remember these things. These are very important. Central dogma is the transfer of information from the DNA to protein. Okay, so DNA to DNA replication, everyone knows. DNA to RNA is the transcription, and RNA to DNA is the reverse transcription. Then RNA is to protein is the translation. Okay, Francis Crick gave the central dogma many times asked in the examination. Okay, so if I am saying that many times asked in the examination or very important, please note it down. This is really going to be important for your examination. Okay guys, so let's go ahead. And cell organelles and their function is very much important in many examinations. So nucleus here is the housing of DNA, and it was discovered by Robert Brown in 1838. Mitochondria, energy production or powerhouse of the cell, and it was discovered by the Colliker in 1880. And Golgi apparatus, sorting, packaging and transport of the proteins, and Camillo Golgi discovered in 1898. And endoplasmic reticulum, synthesis and processing of the protein, lipid expression, it was discovered by the Porter in 1945. Chloroplast was discovered by Schimper, and it has the function of photosynthesis, all new. Flagellum has the locomotion and sensory function, and it was discovered by the Leeuwenhoek. And at the same time, vacuole was also discovered by the Leeuwenhoek, which has a function of storage. Lysosome, very important, was discovered by de Duve with the peroxisome also, and they both have the kind of degradation of the larger and hydrogen peroxide in the cell. So ribosome discovered by Pallad in 1838, and it has the role in synthesis of proteins. Nucleolus is not given here, so nucleolus has the function of producing the what? rRNA. So it was discovered by Felis Fontana in 1774. So let's come to the another topic, that is Hardy-Weinberg principle. So guys, what happened? Hardy-Weinberg principle in the last few years is asked in the examinations. So you have to remember that what is allele frequency, what is gene pool. this is a vast topic, so please go and read about it. Okay, but this is the enough information. You can remember the formula p square plus 2pq plus q square equal to 1. That is in the form of a plus 2b plus a square plus 2b plus b square equal to 1. So bb becomes what? Bb becomes the homozygous, heterozygous and the homozygous recessive. So you can go with the table which is written here, p square aa pq, and then what happens in the cross, you are getting the 2, you can say heterozygous, so 2ab. Okay, so 2pq. So if suppose they are giving you the frequency, then what you have to do, you have to get the number of the offsprings. Then you are going to get the proportion of how many like kind of like affected, or you can say how many kind of characters are present in that organism by deducing the allele frequency into offsprings, or offspring to allele frequency. Okay, so let's discuss ahead, because it is a vast topic. I'm not going to discuss more about it. Okay, so molecular technique. In molecular technique, basically discoveries becomes very important. PCR is was discovered by the Carey-Mullis, and it has a DNA amplification, all know. Northern blot was discovered by Alvin, Kemp and Stang, and it has the quantification in RNA. So western blot has the same for protein, it was discovered at Tobin, and southern blot by Southern, and for DNA. RNA interference was discovered by the Fire and DeMello, and it has the role in gene silencing. DNA fingerprinting by Alex Jeffries, and it has like role in suspect finding by VNTR, variable nerve tendon bridge. Okay, so monoclonal antibody was discovered by Milstein and Kohler, and it has role in DNA protein interaction. Okay, sorry, this is wrongly written here, DNA protein interaction. Basically monoclonal antibodies are for like various clinical implications. Okay, so DNA polymerase was discovered by Kornberg, and it has role in replication. If suppose DNA protein interaction is written here, so I'm going to highlight what actually DNA protein interaction is detected by. So DNA protein interaction is detected by DNA footprinting. So it is a very nice method. If it is written here mistakenly, then take it as an advantage, and just remember that DNA protein interaction is detected by the DNA footprinting, not the fingerprinting, footprinting. Okay, so lac operon discovered by Jacob and Monod, and it has role in gene regulation. And DNA sequencing was discovered by Sanger, most of you know, and gene sequencing is the main part. Recombinant DNA technology was discovered by Cohen and Boer in the like in vitro. Okay, so it has a role in gene cloning. So cloning vectors and their insert capacities are very important. So vector system, plasmid has 0.1 to 10 kb kilobase pair, bacteriophage lambda 10 to 20, cosmid 35 to 45, bacteriophage P1 80 to 20, BAC 50 to 300, then the bacteriophage derived AC P1, and it has 100 to 300, YAC has 100 to 2000, and more than 2000 in the human, that is HAC. Okay, so sources are given here, which are the like kind of sources for these kind of vectors. So let's go ahead and discuss the blood. So blood has different corpuscles or cells. So erythrocytes, basophils, lymphocytes, monocytes are of two types, basically dendritic cells and macrophages. Okay, so macrophages are derived from monocytes, many times asked in the examination. Platelets, neutrophil and eosinophil. So you can here look into the nucleus structure. Okay, basophil is bilobed, neutrophil is multilobed, eosinophil is also bilobed. Okay, so you can remember these kind of things, bilobed, and while doing the estimation of these like number of the, or you can say distribution of these corpuscles, white blood cells are basically counted on the, you can say, marginal part, and RBC in the central part of the Neubauer's chamber. Okay, so this is called the Neubauer's chamber. So calculation is given, you can go through it if you want. Normal ranges of the RBC in male is 5 to 5.5 and female is 4.5 to 5, and newborn 6 to 8. Remember, neutrophil has the distribution of 50 to 70% of the like blood cells, and of the leukocytes, and monocytes has 2 to 8%, eosinophil 1 to 2%, basophil is 0 to 2%. Okay, so you can go through the absolute value also, but absolute value are not asked, but they can ask what is the absolute value for the leukocytes. So it is what, it is 10,000 to 12,000, 8,000 to 12,000 basically, and varies among them in the range. Okay, so some diseases are there, and pathophysiologies are given here. So Alzheimer's, amyloid beta, and tauopathy, remember please. And Parkinson's disease has low dopamine concentration in the brain, basically substantia nigra. And Huntington disease has CAG repeats more than 40, or basically more than 200 also. So Cushing's syndrome, high level of cortisol, goiter, thyroidism. Please remember hypothyroidism. Okay. So Addison's disease, hyposecretion of glucose and mineralocorticoids both. So they can confuse you by giving the single option. Okay, so they both have the hyposecretion. Klinefelter syndrome, why? What happened here? XXY, 1X extra. So Down syndrome, trisomy of 21, it is also called Mongolism. And Patau syndrome, it has trisomy of 13, and Edward has trisomy of 18. E8 Edward. In this way you can remember. You are never going to then forget these things. Okay, Turner syndrome has X0, that is only one X. Cradut syndrome has broken chromosome 5. Okay, so let's go ahead. Lenses and mirrors becomes very important, guys, for the examinations. And the lenses, you can think about the converging lens is the convex lens. How I remember, I'm going to tell you. Converging lens, CONV, conv, and CONV, con for convex. Okay, so convex and converging, please remember these things. Okay, and about mirror, the things are opposite. Convex mirror has the diverging, and the concave mirror has the converging. So converging and diverging lens, basically you can remember in this way. So this was the trick which I wanted to share you. Then the vision problems become very important. Myopia is basically what? Nearsightedness, in which you are going to see the near object, but you are not going to see the far object. Opposite with the hypermetropia. In the hypermetropia, that is farsightedness, you are going to see clearly the far objects, but not going to see the nearer objects. This is basically in the adult people. Okay, so astigmatism. So what type of lenses basically are used in these kind of persons? So you can see in the upper panel that the image is formed in the front, or you can say in the earlier part in the myopia. Okay, and in the hypermetropia, the object image is basically formed beyond, okay, or far from the retina. And in the astigmatism, what happened? Orientation was basically altered. So you can see that the cure of all these kind of the vision problems basically are done with the lenses, not the mirror, lenses. So convex lens is used for the myopia, and you can see that the image was formed at the retina itself. And then hypermetropia, convex lens was used, and astigmatism has the kind of combined lens. You can see that the convexo-concave lens is used in the astigmatism. And one more thing, presbyopia, which is seen in the aged people, basically is cured by the cylindrical lenses. Okay, you can remember in addition to all these. So guys, microscope parts, becomes very important for the AIIMS PhD entrance. Okay, so you can go through all the parts from head, arm, mechanical stage, then the coarse adjustment, fine adjustment for finding kind of thing, brightness adjustment is there, light switch, illuminator, then the condenser, diaphragm is there, aperture is there, stage clip, objective lens, okay, nose piece, objective lens, and eyepiece. Eyepiece is the ocular piece, basically ocular lens you can say, from where you are going to see. And objective lens is the lens nearer to the object which you are going to see. Okay, so you can go through this thing. This thing is very important. Many times in the AIIMS entrance examination, like 3-4 times they have asked these things. Okay, so basically in this kind of figure 1, you are going to focus on the magnification, color code, numerical aperture, okay, and the tube length, And if you are much more interested, you can go through the aberration correction. Okay, what is the aberration correction and flat shield correction, lateral magnification is there. So magnification divided by numerical aperture all the time, not the focal length. They are going to confuse you with the like kind of things, focal length, everything they are going to give you. So please mark this 1.40 oil which is written here. So 1.40 is the numerical aperture. These two things are very important, 1.40 and this 0.21 which is the working distance. Okay, tube length is given here, you can remember or remember not. So three things are very important: numerical aperture, working distance, and magnification color code you can remember, and the basically this 0.1 the cover slip thickness. Okay, so these four things please remember. So guys, let's discuss about the coronavirus. It is a very hot topic and questions can be asked from these kind of hot topics. So family Coronaviridae, as you all know, and it belongs to the what? SARS family, severe acute respiratory syndrome. So this belongs to the SARS family and MERS family. These are the two like kind of Coronaviridae, say these are the two genuses. Okay, so from this is not the new thing. In 2002, South Asian countries have felt this kind of pandemic in 2002, where 15,000 to 20,000 people were affected, and it has the RNA as genome. Very important, they can ask you, this is a retrovirus. Okay, so spike protein as the vaccine target. The protein which is targeted for the vaccine formation is the spike protein. So envelope, you can see here in this structure, envelope, small membrane protein is there. Okay, membrane protein M is there. The nucleoproteins are in the inside part, genomic RNA is there, and the hemagglutinin esterase. This is very dangerous thing. This is basically the toxic part of the virus. So please remember this thing, hemagglutinin or hemagglutinin esterase. Okay, so guys, this was all about the lecture. I hope you have enjoyed the video, and please guys, please this is my sincere request and humble request that please share the video. I'm working day and night for your like kind of benefit. So guys, please, it is your responsibility to like share the video to your nearer ones, and share this video to your juniors, your colleagues which are going to give the examinations like CSIR JRF, ICMR JRF, DBT JRF, and GATE. And this lecture series is basically built for the AIIMS PhD entrance. So AIIMS PhD entrance is also fine for them. Okay, so the only one lecture which is remaining after this is the interview wala preparation. So I'm going to prepare you about the interview of the like AIIMS PhD entrance, and it will be exclusive on the Microscope India, and I'm going to take that video on like the 30th or 31st. So 31st is your exam. All the best guys, please do best. Please think and then answer. Why? Because there is the 1/3 negative marking. So you are not going to do all the things in hurry. Please check if you have done like right or not. After opting, you have enough time at the end, then please go and make a run through. This is the best thing. Okay, so guys, it was all about the tips and everything. I have covered the 12 curriculum lecture basically from the like kind of aptitude part, and the only one lecture which is remaining is the like interview preparation for the 8th November departmental assessment. Okay, so in between I'm going to take, if I'll get the time tomorrow, then I'm going to take the video tomorrow, or otherwise it will be after the Diwali, like 5th or 6th. Okay, so guys, all the best to you all. Thank you so much for your kind attention. Thank you guys.